Oh, it's not going. It's not doing the video. Don't worry. That would be weird. You guys just sitting back there. All right, all right, all right. So let's do that. There, share. Do you guys want to do teams this time, or do you want to just do individual again? Any preferences? I think individual. Okay. Test yourself. Yeah, yeah. I get it. My computer is super quiet. Let me try to get that back one. Pretty good. Kind of cut out just for a second. Sorry. Not my phone. There it goes. All right, are we ready? All right, so this one is going to have slides in between, so I can like talk about some of the stuff whenever we get through it. Anyways, first up, we're going to, we need to know about safety. So we're going to go through the sections of the concepts you need to know. So you will need to know about safety. Just obvious stuff like the stuff that's on the paper that's posted online I'll show, but put your things where they belong and all of that. So here is, I think there's only one question from safety. Where should broken glass be disposed? All right, so yeah, so good. Got that one pretty good. All right. With the stains, now I'm going to get into each of the stains, but with the stains, you're going to want to know the steps, right? So like if we're looking at a gram stain, you'd want to know what I mean is, you want to know heat fix your slide, then crystal violet, then the iodine, and then the alcohol, and then the safranin, and then you look at it. But there's rinses in between. Yes, there's rinses in between. That's not quite as important. And you don't need to know the times. I don't need you to know that it sits for a minute, or anything like that. More just the stains and what each one is used for. So yes, so each stain that's in those stains has a purpose. So whether we're talking about the acid fast or we're talking about gram stain, each stain is used for a specific reason. I would be aware of that. Positive and negative stains. Remember that cells have a negative charge. So if it's a positive stain, it's going to stick to the cell itself. If it's a negative stain, it's going to be repelled by the cell. And that's why we see the background getting stained. Okay, and then also be aware of what would happen if you skipped a step in that process. Because that is, I know that's on at least one of the questions. What would happen if you skipped this step? What would your cells look like? All right, next thing. All right, you can select more than one for this one. What is the purpose of heat fixing? You can have more than one purpose. Let's see if we can find it. All right, so our answers here are going to be adhere to this slide and kill it. I'm actually really proud of you guys for not too many of you choosing the stain it, because a lot of people do. It's preparing it for the stain, but it doesn't actually stain it, right? So good job. All right, what is the counter stain used in the gram stain? Right, so our counter stain. The counter stain is sort of like what you stain the cells with just to be sure that you can see what's going on afterwards. So it's usually the last stain after. So with acid bath, it's the methylene blue, right? We stain pink with the fuchsia, the carbon fuchsia. Anyway, so for this one, for gram stain, it's saffron. It's just going to give us some contrast. All right, what color will a gram-negative bacteria be after the gram stain? Gram-negative. Yes, it should be red. More easier. All right, what is the first stain used in the capsule? All right, so this one is the Congo red for that one. It's the one that gave us that we spread it across and let it dry and then drop the medieval stain on it. But Congo red, it is a negative stain. Cool. All right, this can have more than one answer. What type? I just said it. What type of stain is Congo red? It could be more than one of these. How many of you guys are listening? Alrighty, so yeah, that's going to be a negative stain, and it's also going to be a background stain. They just kind of go hand in hand. Where did my cursor go? This is the thing, is if I move even like an inch, there it goes. All right, this is a pure culture. What stain is pictured? Yeah, so this one is going to be the endospore stain. So the little endospores are staining that bluish greenish color, and then the actual vegetative cells and cells around it is going to be the red from the saffron. Watch it be the next question. Watch that be. All right, what is the term for the red structures in this image? I did say it. Let's see if you're listening. Yeah, so that's going to be vegetative cells. The cells around the endospores are called vegetative cells. Well,
was there more than one answer to that?
No, there was just. Did it have more than one?
Yeah, it said endospores.
Yeah.
Oh, well, that shouldn't have had more than one. It should have just probably just did get selected correctly. All right, which of the following bacteria would be acid fast positive? All right, yeah, so this is going to be the Mycobacterium blei. Acid fast The only ones that are acid fast are going to be mycobacterium. All right, what is the primary stain used in the acid fast stain? So our primary stain is the one that is used first, typically. This is the carbofuscin in the case of the acid fast. And it will allow it to bind to the waxy, mycolic acid. In which staining procedure did we use steam? Yeah, so that is the endospore stain. Which of the following structures allow for bacterial motility? Select all that apply. Well, okay. Well, that's lucky for you guys because it is only one of them. So if you pick the right one. All right. So for bacterial motility, we have only flagella that allows us to move around. All right. So bacteria do not have cilia. That's like they don't have that at all. Fimbriae for attachment and pili, the sex pili for transferring during conjugation, right? So that's that only flagella for movement. Now the eukaryotes do use flagella or do use cilia sometimes. All right, so here are some of the stains that we would expect you to know. Simple stains, gram stains, acid fast stains, capsule stains, the wet mount, and the endospore stain. So those are the ones that we went through in the lab. I would know what their purpose is, what the purpose of each stain is, what the purpose of each step in each of these is, all of that stuff. Maybe I would like make a card and just like write all of it down as much as you can on that. And that would allow you to kind of get that information like set up somewhere useful. I don't know, organized, I guess that's the word most people would use. All right, so we just did the ELISA, but now we're going to go through the ELISA stuff. What does the E stand for? All right, so yeah, the E is going to stand for ELISA, or enzyme. It stands for ELISA, guys. Enzyme. You guys got that. Now I lost my thing because I was stupid. There. All right, what does an indirect ELISA look for? All right, so yeah, so at least you guys are getting it both in the right direction. So the indirect, that's the one that's going to be looking to see if the patient has the antibody. The direct is the other one. We've got that. All right, so now the principles of how it works. Remember, it's going to change colors due to that enzyme cleaving a substrate, right? And that enzyme's only there if it's positive. You want to know about what antibodies and antigens are, general concepts here. Indirect immunoassays versus direct immunoassays. Because these terms don't just apply to ELISA, they apply to like a lot of testing that's done. So that'll come up again later on when we're talking about diagnosis of infections in this next set of chapters. All right, how the color change happened, right? I just said over that. And then rapid testing. All right, for the DNA stuff. Yes, the online stuff and the transformation simulation, these are things that can be tested over. I would Well, let's just go through this part first and see what I'll tell you guys to do. So what does PCR do? Yeah, so what's gonna be going on with PCR, we're gonna amplify DNA samples. It has nothing to do with antibodies. That's the kind of thing that ELISA's gonna deal with. PCR, polymerase chain reaction. What kind of polymerase? DNA polymerase. Okay. All right. Which DNA fragment is the largest in the list? Largest. All right, yeah, most of you guys got that one. That's gonna be the 35 kilobase pairs or 35,000 base pairs. All right, which of the following DNA fragments would travel the farthest on an agarose gel? Yeah, so it's gonna be the smallest one. Good job. All right, what type of cells are capable of taking up exogenous DNA? All right, so the answer we are looking for here is competent. If a cell is competent, that allows it to take up exogenous or outside DNA pieces. This is what we did in the transformation experiment. So those Tupperware were competent cells, and they don't have to be E. coli. All right, so with the DNA stuff, I want you to know about DNA structure and function. We kind of already learned about that with the test, I wouldn't really worry about that too much on this one, I'm just saying. DNA fragment sizes, I would be aware of. Gel electrophoresis, how PCR works in a practical sense. So note steps, essentially, the general steps. So remember, it's going to be, and this was in the lecture test too, but you carry it over. The heating being the denaturing step to split it apart, and then the annealing step, and then the elongation step, right? So, all right, we have bacterial transformation, the steps that are involved there, and the results that we might see on each media type. So what would you expect to see on the ampicillin only, for example? And then plasmids, what are they? Just to be clear, I know you guys probably know this. You guys have been pretty good about this semester, but usually that's one of the most commonly missed things on the test. If I ask you what plasmid is made of, most people say protein. It is DNA. So just be aware of that. It's DNA. It's carrying genes and stuff. All right, moving on to the eukaryotes. You do want to review your drawings and just keep them kind of fresh in your mind, and how to recognize some of these guys. Which organism is this? That's pretty much how the test is going to be like. Yeah, so this is penicillium. I do appreciate that you guys guessed, similar organisms, the fungi, and that part is, crucial. But yeah, that's penicillium, that particular one. That could be one or the other. All right, what is the name of this organism? All right, so that's Giardia. I'd be aware of that one. Hint, okay. It's pretty recognizable though, so I don't feel like it's too bad. All right, the eukaryotes, you want to look over those organisms you had to draw, the important structural and reproductive differences, and then that crap that you had to write about like the life cycles and stuff like that, that stuff's going to be tested on the test too. Tropozoite versus cyst, the concept of that, and then identifying things based on differences. That's it. I mean, I don't like the eukaryote part, but all right, bacterial growth, growing things on bacteria or on media. All right, which is not a term used to describe colony morphology? Colony morphology. People forget about this one, about colony morphology. It goes back to days. All right, so colony morphology is just referring to terms that are used to describe the actual literal growth on the plate. And so the one that it doesn't fit in this, that's hyphae. Hyphae has to deal with stemming off of mold, making it look fuzzy. That's not really the colony morphology. That's just the structure. All right. What is the name of this type of culture? Yeah, so that's the slant. Most of you guys got that one. It's kind of hard to see it. But if, of course, in real life, you guys would be able to pick it up. You get an advantage with that. How was this organism isolated? Yeah, good job, guys. This is one thing that almost everybody just takes with them is that freaking street play. Like nobody forgets it. All righty, cool. All right, which arrangement of bacteria do you observe in the picture? Out of your choices here. So our arrangement has to do with how the cells are actually, shaped out, like working together. That's really, I'm not describing that well at all. How they are arranged with one another, amongst one another. So that was strepto. Strepto has to deal with the chain. We saw the chains of bacteria forming in that picture. There was in the picture like two that were like this right next to each other. That technically we could have put diplo, but I hate that because clearly this is a strepto organism. And that would have just been two that just were hanging out on their own off of the chain, like not diplo. So that's a different deal. Anyways, don't like that tripping you guys up. So yeah, that's just going to be strepto for this. Strepto. I remember. All right, what is the name of the shape of these bacteria? The actual shape of the cells here. It's a little hard to see them on this screen, but it is easier on that side. All right, so this is spirillum, basically spiral shape or general spiral shape. You guys will not be seeing any slides with that. It's only going to be bacilli or cocci, so I'd be aware of those. Right, colony morphology, what that stuff looks like when it's growing on there, at least the words that are appropriate to describe it. The different kinds of media, like we're talking about differential versus selective, because we did talk a little bit about that. We're going to actually do that whenever we get back. I feel like we're going on sort of vacation. But, anyways, we have colony morphology, media types, isolation techniques like the streak plate. There are other ways to isolate organisms as well, like the serial dilution, stuff like that. We have cell shapes and cell arrangement, not to get them too confused with one another. All right, the microscopes and how to use them properly. If you observe a specimen at 40x objective and have a 10x ocular lens, what is the total magnification of the specimen? So this is going to be 400. So you just multiply it for those that don't know. Which objective lens do we use for oil immersion? Yeah, so that's a good job. All right, so you want to be able to calculate magnification, understand how the microscope operates, know the parts. I will let you guys know that there will be a microscope that will have a part labeled, at least one part labeled, that you will be asked about what it is called and what it does. Then understand why we use oil immersion. All right, next is the Kirby-Bauer assay. This is our antibiotic susceptibility and resistance assay. So which antibiotic is this bacterium least susceptible to? Which one is it the least susceptible? All right, so our answer is going to be E for this one. So you guys got that one, right? Because it was clearly there wasn't stopped at all by that. This was not, didn't care. All right, so I know what a zone of inhibition is. The minimum inhibitory concentration, that's the MIC. We can calculate that from the zone of inhibition. The Mueller-Hinton auger, remember that has to be kind of all even. We want pH level maintained as well. Same thickness between plates. Then we have the terms about bactericidal versus bacteriostatic. All right, so that is that for that review quiz. If you guys have any questions about any of that material, let me know. And then, yeah, have fun studying, I guess. This one should be a little bit better, especially because a lot of the stuff we've already seen in the lecture. So. Okay, good job, good job. Yes, it's going to be on Scantron.